Alrighty. So this is a relatively small group. You can go on the next one. This is under Objective 1, 1.1.3. And this is a small group of people. And mostly what we're looking at is trying to dig in a little bit more on what's going on after, or what kind of effects rootstocks in particular are having on the behavior of the scions and the quality of the fruit after harvest, particularly solanaceous being tomato and pepper, and then cucurbits, primarily watermelon, a little bit on melon. Okay. Under the solanaceous, okay. This is the first summary of work from a master's student that Joe Sharons was chairing at Ohio State. And the idea here was to look at bell pepper grafted onto a hot pepper rootstock to make sure that there was no negative effects of the hot pepper, for instance, transmitting capsaicin into the bell pepper, as well as screening for the usual quality traits. So this was an Aristotle, and then a number of the scion treatments were used. And then the variables looked at were the usual things that we associate with yield, which would be fruit weight, and then quality, which would be load count, carpal wall thickness, which has to do with retention of firmness, also the shape index, and then quality aspects such as soluble solids, acidity, and capsaicate. There was also a consumer sensory evaluation in 2017 to follow up on this. And then the other study that was done was using Aristotle and Carolina Wonder and narrowing it down to just 3 rootstock treatments. Okay, next slide. So basically what happened here in the first one in 2016 was there was very little difference among the rootstocks on the quality of the bell pepper. The only thing that was occurring was a little bit of difference in the shape index, and the carpal wall thickness was actually a little bit thicker when you added the rootstock component to it. The lobe count was also slightly changed, not enormously. I don't think terribly negatively. You still had 3 to 4 lobes, which is what you'd want. And then in the sensory evaluation in 2017, they basically were unable to tell a difference between one that was grafted on a hot pepper, a bell pepper grafted on a hot, versus a regular bell pepper. It was about evenly distributed, which is good news in terms of not being able to pick up any extra heat from a bell pepper that would be unexpected. And then the root treatments also were about the same as far as liking or not liking. They were about this evenly distributed across the root treatments. The soluble solids content was just slightly shifted with the harvest date primarily, and was generally around 5, which is pretty normal for bell pepper. Next slide. So the conclusion of this, which is kind of, one of those few, moments, is that when you put a bell pepper on a hot pepper rootstock, you're not going to get an adverse effect on flavor or shape. And the next component, of course, that I know Dr. Sharons is working on is going to the next level, which is going in reverse. What happens when you put a hot pepper on bell pepper? And I think that will be occurring in the next couple, well, this year and next year. Next slide. So now I'm shifting over to some work that was done on tomatoes with Shin and Jeff Brecht at Florida. And this started out as a high tunnel experiment that kind of morphed over into a grafting grant when this one ended. The first part of this was looking at how this particular variety did under high tunnel and open field in terms of shelf life response and quality aspects, and essentially there was very little effect on the quality. Next slide. This is not unusual with tomato. So what they were doing here, and they're continuing this work now in a new study, looking at, in this case, they're switching over from a normal or average large-sized tomato into the grape size to see if they can be more easily evaluated for effects, especially on number of fruit per plant and things like that, and a lot of quality aspects that go into tomato as well. soluble solids, pH, carotene, lycopene, vitamin C, antioxidants, and phenolics. And also the storage, the shelf life studies, and consumer sensory analysis. Now, the problem with tomato is because it's generally grafted on tomato, it's much less likely to see strong positive or strong negative effects. And it'll be interesting to see that using essentially in this case, what they'll be moving into with the grape tomatoes would be the indeterminate types as well as determinate. So it may be that indeterminate may show a different effect from what's been normally used with the larger ones, which are primarily determinate types. And then also consumer sensory analysis. One of the other problems with grafting is that sometimes you'll get a consumer test that says, yeah, there were good effects, sometimes it's a negative effect, and of course the worry is always that the negative effect may have a huge impact on whether people want to adapt this technology. Let's see, next slide. Okay, that's a quick wrap up on tomatoes. So we have a student that will be continuing on the pepper work. We have a student in Ohio, another one in Florida, or maybe two working on the tomato side of it. Switching over again on the cucurbits, melon, in this case Gallia, has been quite the challenge for sensory tests when it's grafted. This one in particular is the one that worries me, at least, as far as adaptability of cucurbit grafting, because this is the one that seems to be very sensitive to the year it's grown in, the rootstock that's used. The one common thing is that Gallia seems to be very, very sensitive to what's going on. And Shin will be continuing this work looking at how Gallia sensory analysis and quality changes in response to rootstock. Okay, next slide. Okay, and then switching over, I have a student working on this. Marlie's working on watermelon effects in response to rootstock. And unfortunately, seeing people here, thing didn't happen right. Anyway, one of the problems with rootstocks, we talked earlier, they're talking about using bottle gourd as a potential means of getting rid of powdery mildew on the scion. We see varying effects with bottle gourd and interspecific, which is Machado times maxima, on quality of watermelon. And in this lower corner, what you see is non-grafted watermelon and one that was put on snake gourd, which is a wild watermelon. I want to say wild watermelon, it's actually a gourd. So you have to choose your rootstock with care. It's very clear here. This hollow heart syndrome, along with a really poor color, indicates that there's a different growth rate going on in vigor. The other problem that we've had over the years with seedless watermelon in particular is that when you start growing seedless, you tend to have more hollow heart. And there's something called density. You have different aspects of density going on that indicates that you could have less dense melon would have more hollow heart. And this many times is screened at the seed level. And then there's the whole interplay of is too much water, environmental conditions, or pollination all interacting with this. So one of the problems with rootstocks is, of course, for watermelon is where does it cross the line between yield and quality? And one of the more obvious quality choices here is hollow heart. Next slide. Okay. So, whoop, somehow we missed. Okay. I guess what it was is it overlaid. I'm sorry.
You want that one or the other one?
No, the other one. Yeah. I must have had a two place on there. So hollow heart starts out as this very small crack in the middle, and it gets progressively by the center. That's no longer considered marketable, and then you can put your fist through the last one. And obviously, this is a negative in terms of what a grower or possibly a distributor wants. Some people argue that when you have this relatively dried out flesh, that it's sweeter, then it's more crunchy. But the key here is that overall, this is something you either are going to have to make sure you don't deliver to a consumer, or you want to really figure out what might be going on relative to the grafting aspects of it. And what we found in the past is that occasionally, depending on the year, the type of rootstock that's used may minimize the incidence of hollow heart. 2 years ago, we were working with Richard Hassel finding just by chance that watermelons that we had picked up from him, that we were having much less incidence of a hollow heart that year, and it was a really wet year, than we did with our non-grafted melons. The next year we didn't see this effect again. So we decided to drill down a little bit more this last summer. Next slide. And the idea was to look at how this would happen if we used a scion that we knew was very sensitive to hollow heart, and we're grafting it onto different types. We use emphasis, which is the bottle gourd, and then carnivore, which is interspecific, and also a non-grafted. And sure enough, well, there's a pollination part of it overlaid on this, but primarily what we were able to demonstrate was that if you want to induce hollow heart, you use Liberty for sure. So that's good news if you want to pursue this. And the other thing was that in this particular case, we were able to show that carnivore did indeed reduce the, which is right here, did indeed reduce the amount of hollow heart compared to the bottle gourd or the control rootstocks. So that was good news for us. Next slide. And then we also were able to show that tissue was firmer from carnivore and also from Kazako, which is interesting. This is another thing that comes up a lot. About every other study that's run on watermelon is interspecific hybrids seem to increase the firmness of watermelon flesh. And again, we're not really sure why. And part of the study that Marley was doing initially was to do all of these massive measurements, I think which she went through 500 or 600 fruit, and also then to start drilling down into what might be going on at the cellular level. And in this case, what was interesting here was that hollow heart flesh was actually softer, at least the way we were measuring it, which was with a regular penetrometer system. We weren't using shear force, something like a Kramer shear press. We were using a regular penetrometer bit. The next slide, please. Okay, the other things that Marley was looking at were the health benefit components that go into watermelon. In this case would be lycopene and also the amino acids citrulline and arginine. And in this case, there is no difference in lycopene content for either rootstock or the hollow heart presence or absence, those melons that had it or didn't have it. The same was also true for citrulline. I did see, even though it wasn't statistically different, I did see a little less citrulline present in carnivore watermelon, which is something that I tend to see a lot, that the firmer watermelons do tend to have less citrulline. Arginine, in contrast, was a little bit higher in the non-grafted watermelons than it was in the grafted, but again, there was no difference in hollow heart. And these are all from heart tissues. This is to me the easiest way to keep consistency going across a number of samples. Next slide. Now, in the case of the soluble solids content, a simple refractometer measurement, it was lower in carnivore and the interspecific rootstocks, which is interspecific, I'm sorry, and also in those with the hollow heart. pH was actually higher in hollow heart, which I generally assume is an indicator of ripeness, but in this case was still well within the pH ripeness zone of around 5.5. And also a little bit higher in the lagenaria, the emphasis rootstock or bottle gourd. And we looked at the sugar composition as well, and what we found was that the total amount of sugar was actually highest also at emphasis, which is bottle gourd, which is kind of surprising to me. I would have expected to see it more in carnivore. And then also of the sugars, glucose was highest in this particular rootstock with very little difference in other places. Next slide, please. And so the next, or part of this, was to also look at the cell structure itself. And so Marley did confocal work to try and figure out the area of the cells. And as would be expected with hollow heart, and has been reported using light microscope as well, as there are larger cells with hollow heart. And that may explain a bit more of what might be going on as the hollow heart develops. And there's a theory that there's different growth rates among cells, and that's why when you have large cells, they're that means they're growing at too fast a rate, and they're more likely to pull back and cause hollow heart. Surprisingly, also emphasis had larger cells than carnivore or the control. I'm not really sure exactly why that would be, except perhaps carnivore is somehow making more dense cells. This is something that we'll probably look at again in the next slide. Okay. Oh,
One or the other one?
sorry, keep going. So the next on the list for Marley to do is looking at the cell wall composition itself in these hollow heart and also in these grafted ones. Trying to figure out if there's some difference in the carbohydrate structure or the types. There's been some work that indicates there's no difference in total amounts of soluble or insoluble type pectins or cell wall material, but Marley has a chance to look at the actual carbohydrate composition in this to see if there's anything that would indicate there might be some sort of effect that's being conferred by this interspecific rootstock, and particularly that might be associated with firmness, to start trying to explain why we get this effect. Okay, next slide. That's it. Okay, so that's where we are right now, and we're, I would say, pretty much on target where we need to be as far as what we had outlined in the grant. And I think I'm open for questions, and I probably caught us up on time.
Thank you, Penny. Yeah, let's open it up for any questions. All right, you know, the biggest thing that strikes me is the question, to what extent can we, you know, with this cross-disciplinary group, and just to have people think about this, is, you know, one of the main things that you're showing is that we can add value to products.
Right.
Whether that's life storage, longer time in the field, or actually human health benefits. You know, for maybe the advisory committee, what are the value added benefits, and then is there a market that can help drive that? So that's a thing that pops to my mind in terms of maybe getting some feedback on. All right.